Welcome to the infection tube. Have you ever wondered what happens when a pathogen enters our body? How does our immune system recognize it and deal with it from the beginning to the end of the fight? Today, we'll explore this incredible process in detail. Let's get a pen and paper and have a session together to explain everything from beginning to end. First and foremost, our immune system must recognize the pathogen. So the first question you need to understand is, how does the immune system recognize pathogens? Now, this is the pathogen, which may be a bacterium, a virus, or a fungus. And this is our immune cell, macrophage, for example. The pathogen carries specific molecules known as Pathogen Associated Molecular Patterns, call it PAMPs, present on its surface or inside it. Pathogen Associated Molecular Patterns are structural components of pathogens. Common examples are lipopolysaccharides, found in the outer membrane of gram-negative bacteria; peptidoglycan, found in the cell walls of gram-positive bacteria; flagellin, found in bacterial flagella; double-stranded RNA, found in many viruses; mannins and beta-glucans, found in fungal cell walls. These all are structural parts of different pathogens that act as pathogen-associated molecular patterns. On the other hand, our immune cells, like macrophages, have pattern recognition receptors that detect these pathogen-associated molecular patterns. Examples of pattern recognition receptors include toll-like receptors, nod-like receptors, and Rig-I-like receptors. Actually, all pathogens have pathogen-associated molecular patterns in one way or another. Do you know which cells express pattern recognition receptors? A variety of immune cells, particularly macrophages, dendritic cells, neutrophils, epithelial cells, and B cells. The role of pattern recognition receptors is to recognize the pathogen-associated molecular patterns. For example, TLR4 on immune cells recognizes lipopolysaccharide from the outer membrane of gram-negative bacteria. NOD2 recognizes muramyl dipeptide, a component of bacterial peptidoglycan. TLR5 recognizes bacterial flagellin. TLR3 recognizes double-stranded RNA from viruses. C-type lectin receptors like dectin-1 recognizes beta-glucan, and dectin-2 recognizes mannin in fungal cell walls. Not only do pattern recognition receptors recognize pathogen-associated molecular patterns, but they also bind to them in a lock-and-key fashion, forming a PAMP-PRR complex. Once this complex is formed, it performs two very important roles. It opens two locks: 1. Initiating phagocytosis, and 2. Activation of immune cells. First, let's talk about how it initiates phagocytosis. When pathogen-associated molecular patterns bind to pattern recognition receptors on phagocytic immune cells like macrophages and dendritic cells, it triggers signaling pathways that prepare the cell for phagocytosis. This involves cytoskeletal rearrangements that enable the immune cell to engulf the pathogen into a structure called a phagosome. The phagosome then fuses with a lysosome to form a phagolysosome, where the pathogen is degraded by lysosomal enzymes. As the pathogen is broken down in the phagolysosome, additional pathogen-associated molecular patterns may be exposed and recognized by pattern recognition receptors within endosomes or lysosomes, further stimulating cytokine production to ensure a sustained and effective immune response. Now let's focus on the activation of immune cells. The binding of pathogen-associated molecular patterns to pattern recognition receptors also initiates signaling cascades that activate transcription factors within the immune cell. These transcription factors lead to the production and release of cytokines and chemokines, which recruit and activate other immune cells, promoting inflammation and enhancing the overall immune response.
Wow, Dr. Max. Isn't it amazing how our body starts a complex and coordinated response just from recognizing a pathogen? It's like a perfectly orchestrated symphony where each player knows exactly when to come in. The way these signals are sent and received is crucial for mounting an effective defense. But Dr. Max, what exactly are cytokines? And what is their role in coordinating such a robust immune response?
Great question, Sela. Let me explain. Now, what are cytokines? Cytokines are small proteins secreted by various cells in the body, particularly immune cells, that act as signaling molecules to regulate immunity, inflammation, and hematopoiesis. They act as messengers, enabling immune cells to coordinate their responses to infections, inflammation, and other immune challenges. They are numerous, and there are many ways to group or categorize them. We have, for example, interleukins, tumor necrosis factors, interferons, colony-stimulating factors, chemokines, and transforming growth factors. One fascinating aspect of cytokines is their variety in naming. One nice trick here is that the types and names of cytokines often reflect their primary functions, making it easier to understand their roles in the immune response. How is that? For example, interleukins means between leukocytes, reflecting their role in communication between white blood cells. There are many types of interleukins, such as interleukin-1, which promotes inflammation, fever, and the acute phase response, and interleukin-2, which stimulates T-cell proliferation and differentiation. The name tumor necrosis factors reflects their ability to cause tumor cell death. We have tumor necrosis factor alpha and tumor necrosis factor beta. Interferons are named so because they interfere with viral replication. There are type 1 interferons, which include interferon alpha and interferon beta, which induce an antiviral state in cells and activate natural killer cells, and type 2 interferon, interferon-γ, which activates macrophages and promotes T helper 1 cells responses. Colony-stimulating factors are named for their role in stimulating the production of blood cells, colonies, in the bone marrow. Examples include granulocyte colony-stimulating factor, which promotes the production of neutrophils, macrophage colony-stimulating factor, which stimulates the production of macrophages, and granulocyte macrophage colony-stimulating factor, which promotes the production of both granulocytes and macrophages. Chemokines are derived from chemotactic cytokines. It took the first clip from this and the second clip from that, reflecting their role in directing cell movement. This is called chemotaxis. Some chemokines attract monocytes to sites of inflammation, while others attract neutrophils to infection sites. Finally, transforming growth factors are involved in cell growth and differentiation, hence the name transforming. Transforming growth factor beta regulates immune responses, promotes tissue repair, and suppresses inflammation. Understanding the names and functions of these cytokines helps to grasp their crucial roles in orchestrating the immune response, ensuring our body's defense mechanisms are both targeted and effective.
Wow, Dr. Max. It's incredible how our immune system coordinates such a detailed response to pathogens. It's like every cell knows exactly what to do and when to do it. But what happens next? Which immune cells are activated first?
Great question, Sela. The first immune cells to be activated upon pathogen entry are typically tissue-resident macrophages and dendritic cells. Their strategic location in tissues and their ability to recognize pathogen-associated molecular patterns through pattern recognition receptors make them the first responders. Then, other cells are activated by the PAMP-PRR complex either directly, like mast cells and neutrophils, or indirectly, like natural killer cells. Once the immune system is activated and cytokines are released, one of the most famous symphonies in the human body begins to play. It is the symphony of cytokines.
The Symphony of Cytokines? That sounds fascinating. Tell me more about this symphony, Dr. Max.
Sure, Sela. Let's dive into the cascade of cytokine interactions and see how they orchestrate the immune response. This is the PAMP-PRR complex. You must start from here, from pathogen recognition and the formation of this complex, which activates the two forefront cells to release cytokines. This is the starting point. These two forefront cells are the macrophages and dendritic cells. Let's draw them as one cell, and we will name it phagocytic antigen-presenting cells. Why do we use this term? Let's bring one sticky note. This term, phagocytic antigen-presenting cells, helps to clarify the roles of these cells in both engulfing pathogens and presenting antigens to the adaptive immune system. There are other cells that can phagocytize pathogens, but can't present antigens to the adaptive immune system. These are called phagocytic non-antigen-presenting cells, like neutrophils. On the other hand, there are cells that can't phagocytize pathogens but can present antigens. These are called non-phagocytic antigen-presenting cells, like B cells. Understand this? That's good. Now, these two phagocytic antigen-presenting cells produce some common cytokines, and each one also produces specific cytokines that distinguish them from each other. What are common cytokines produced by phagocytic antigen-presenting cell? The first is interleukin-12. It goes to naive T cells to promote their differentiation into T helper 1 cells, and it also goes to natural killer cells to enhance its cytotoxic activity, and to cytotoxic T lymphocyte cells to enhance the cytotoxic activity also. These target cells reciprocally reply by releasing interferon-γ. Let's draw the feedback loop with red color. Natural killer cells release interferon-γ back to the phagocytic antigen-presenting cells. Cytotoxic T lymphocytes do the same, and also do T helper 1 cells. Additionally, T helper 1 cells send interferon-γ to T helper 2 cells to suppress them, effectively saying, "I am now working, my sister T helper 2 cells, and you must rest as we cannot work together."
Dr. Max, interferon-γ seems to play a crucial role in the immune response. What exactly does it do?
Great observation, Sela. Interferon-γ plays a crucial role in enhancing the immune response by further activating macrophages, increasing their ability to kill intracellular pathogens, enhancing the antigen presentation capabilities of antigen-presenting cells, promoting the further differentiation of naive T cells into T helper 1 cells, contributing to the overall inflammatory response, suppressing the differentiation and function of T helper 2 cells, thereby skewing the immune response towards a T helper 1 cells type response and away from a T helper 2 type response. This shift is important for effectively combating intracellular pathogens like viruses and some bacteria. The second cytokine released in common is tumor necrosis factor alpha. It targets natural killer cells, enhances their cytotoxic activity, neutrophils, increases their ability to migrate to sites of infection, acting as a chemokine, endothelial cells, induces the expression of adhesion molecules, making it easier for immune cells to adhere to blood vessel walls and migrate to sites of infection or injury, and the hypothalamus, promoting the production of prostaglandin E2, which increase body temperature, leading to fever, which can help inhibit pathogen replication and improve immune cell function. In response, natural killer cells send back interferon-γ. We already know what it does from the previous explanation about interferon-γ. And neutrophils send back interleukin-1 beta, which promotes inflammation and further recruits immune cells to the site of infection. The third cytokine in common is interleukin-1 beta. It targets neutrophils, chemokine effect, endothelial cells, increasing adhesion molecules, the hypothalamus, producing fever, and the liver, inducing the production of acute phase proteins, enhancing pathogen clearance and tissue repair. In reciprocal, neutrophils send back tumor necrosis factor alpha to further promote inflammation. Notice this fascinating consistency. When neutrophils receive tumor necrosis factor alpha, they can respond by producing and releasing interleukin-1 beta. Conversely, when they receive interleukin-1 beta, they can respond by producing and releasing tumor necrosis factor alpha. It's a symphony of signaling, as we mentioned before, showcasing the amazing consistency of the immune response. Even more impressive, macrophages receive interleukin-1 beta, which they secreted before, and respond to themselves by releasing interleukin-10. What a surprise! They respond to themselves.
Dr. Max, what is interleukin-10 and why do we need it?
Excellent question, Sela. Interleukin-10 is an anti-inflammatory cytokine that helps regulate the immune response and prevent excessive inflammation. It is one of the important calmers of the immune system. Why do we need calmers and regulators for the immune system? They are crucial because they help control and resolve inflammation once the pathogen has been neutralized, ensuring that the inflammatory process does not continue unchecked. Without these calming signals, acute inflammation could transition to chronic inflammation, leading to various inflammatory diseases such as arthritis, inflammatory bowel disease, and cardiovascular diseases. Additionally, they help prevent the immune system from attacking the body's own tissues, which is essential for preventing autoimmune diseases. Other important calmers are transforming growth factor beta, interleukin-4 and interleukin-13, interleukin-1 receptor antagonist, specialized lipid mediators such as resolvin and lipoxin.
So, interleukin-6 is also released in common?
Yes, indeed. Interleukin-6 targets the liver, induces the acute phase response. B cells, promotes their differentiation. T cells, keep this in mind now as we will use it later. Just remember that interleukin-6 talks to naive T cells, and the hypothalamus produces fever. And then there's the calmer, interleukin-10. It reciprocally calms the two phagocytic antigen-presenting cells, as we mentioned before. This type of signaling, by the way, is known as autocrine signaling, where the cell responds to itself. For example, macrophages can secrete interleukin-10, which then binds to interleukin-10 receptors on the same macrophage. Besides autocrine signaling, there's paracrine signaling, where interleukin-10 activates nearby T cells, particularly regulatory T cells. In response, both macrophages and regulatory T cells secrete transforming growth factor beta, which works synergistically with interleukin-10 to suppress immune responses and promote tissue repair, as we discussed earlier in this side block. Now we've finished talking about the cytokines released by the two forefront cells, and it's time to discuss the specific cytokines produced by each one, which serve the primary functions of each cell.
Wow, Dr. Max, this is a lot to take in. Can you summarize what each of these cells and cytokines do for our immune system?
Absolutely, Sela. Let's break it down. Macrophages specialize primarily in phagocytosis and direct pathogen killing, producing cytokines to recruit and activate immune cells. For example, interleukin-8 targets neutrophils, acting as a chemokine and recruiting them to infection sites. Neutrophils reply by releasing interleukin-1 beta and tumor necrosis factor alpha, contributing to a feedback loop that amplifies the inflammatory response. Macrophages also produce transforming growth factor beta, targets epithelial cells, promoting tissue repair. In addition, it has a very important role in determining the differentiation pathway of naive T cells. Transforming growth factor beta encounters interleukin-6 on its way and activates naive T cells to differentiate into T helper 17 cells. However, if transforming growth factor beta encounters interleukin-2 produced by activated T helper 1 cells, it promotes the differentiation of naive T cells into regulatory T cells. TREGs are key producers of calming cytokines in the immune system, crucial for maintaining immune tolerance and preventing autoimmune responses. In response, TREGs secrete interleukin-10 and transforming growth factor beta, which work synergistically to suppress immune responses and promote tissue repair. Dendritic cells specialize primarily in capturing antigens, presenting them, migrating to lymph nodes, and bridging innate and adaptive immunity by activating T cells. To do this, dendritic cells release interleukin-23, which acts on T helper 17 cells, supporting their final maturation and stabilization. T helper 17 cells are initially differentiated from naive T cells in the presence of interleukin-6 and transforming growth factor beta, and they play a key role in combating extracellular bacteria and fungi. T helper 17 cells reply by releasing interleukin-17, which recruits and activates neutrophils and other non-lymphoid cells, such as epithelial cells. In return, neutrophils release interleukin-1 beta and tumor necrosis factor alpha to enhance and amplify the inflammatory response, ensuring a robust immune defense. In addition, dendritic cells specifically release type 1 interferons, interferon alpha and interferon beta, to natural killer cells to enhance their activity, and to T helper 1 cells to boost their mediated immune response, which is crucial for fighting viral infections and intracellular pathogens. In response, both natural killer cells and T helper 1 cells secrete interferon-γ, which we previously discussed its role. Let's talk about the activation of one of the leaders of adaptive immune cells in our body, T helper 1 cells. As we mentioned, this activation happens primarily due to interleukin-12 from dendritic cells and macrophages. Once activated, T helper 1 cells produce interferon-γ, which stimulates macrophages, enhancing their microbicidal activity and leading to better kills of intracellular pathogens such as bacteria and viruses. It also inhibits the differentiation and function of T helper 2 cells, promoting a cell-mediated immune response over a humoral response. This is crucial because the activation of T helper 1 cells, or T helper 2 cells, leads to the suppression of the other, ensuring that both cannot be activated at the same time. This ensures that the immune response is appropriately directed and prevents the concurrent activation of conflicting pathways. The activation of T helper 1 cells also leads to the secretion of interleukin-2, a key cytokine that primarily affects T cells. Interleukin-2 promotes the growth, proliferation, and differentiation of T cells, including T helper 1 cells themselves, autocrine, cytotoxic T cells, regulatory T cells. With the help of transforming growth factor beta, it aids in their differentiation from naive T cells, as we mentioned before, and natural killer cells. Interleukin-2 significantly amplifies the capabilities of natural killer cells, making them crucial players in the body's defense mechanisms. When natural killer cells and other lymphocytes are exposed to high doses of interleukin-2, they can become highly potent cytotoxic cells known as lymphokine-activated killer, name it LAK cells. Lymphokine-activated killer cells have a greater ability to kill a wide range of target cells, including resistant tumor cells, and are capable of targeting and destroying more types of abnormal cells. They also produce higher levels of cytokines like interferon-γ, which amplify the immune response. Clinical applications of lymphokine-activated killer cells include their use in adoptive immunotherapy for cancer, particularly in cases of metastatic melanoma and renal cell carcinoma. Interleukin-2 also plays a multifaceted role in modulating B-cell function, contributing to their proliferation, differentiation, antibody production, class switching, and survival. Activated T helper 1 cells also produce tumor necrosis factor, which promotes inflammation and recruits immune cells, interleukin-3, which supports hematopoiesis and immune cell proliferation, and granulocyte macrophage, colony-stimulating factor, which stimulates granulocyte and macrophage production and activation.
What about T helper 2 cells, Dr. Max?
Now, it is time to talk about T helper 2 cells, which are primarily activated in response to extracellular pathogens, such as helminths and certain bacteria, as well as allergens. Dendritic cells and other antigen-presenting cells present antigens on MHC class II molecules to naive CD4 T cells. The presence of interleukin-4 is crucial for the differentiation of naive CD4 T cells into T helper 2 cells. Interleukin-4 can be produced by various cells, including mast cells, basophils, and existing T helper 2 cells. T helper 2 cells are stimulated by the presence of interleukin-4, and they also produce IL-4. Interleukin-4 promotes B-cell differentiation and class switching to IgE, which are important for defense against parasites and are involved in allergic responses. It also supports T helper 2 cell differentiation and suppresses T helper 1 cells differentiation and function. T helper 2 cells produce interleukin-5, targets eosinophils, enhancing their proliferation, activation, and survival, which is crucial for combating helminth infections and contributing to allergic inflammation. Interleukin-13 targets B cells, similar to interleukin-4, promoting class switching to IgE, epithelial cells, increasing mucus production, and smooth muscle cells, contributing to airway hyperresponsiveness relevant in asthma. Interleukin-10, an anti-inflammatory cytokine that targets macrophages and dendritic cells, inhibiting the production of pro-inflammatory cytokines and reducing antigen presentation to limit the immune response and prevent tissue damage. It also targets T cells, suppressing the activity of T helper 1 cells and promoting regulatory T cell development. Interleukin-6 targets B cells, promoting their differentiation into plasma cells and hepatocytes, stimulating the production of acute phase proteins.
Dr. Max, this is really interesting. It's amazing how our immune system coordinates such a detailed response to pathogens. But what's this MHC class 2 you mentioned? And is there an MHC class 1?
Great question, Sella. Yes, there is an MHC class 1. While MHC class 2 presents antigens to naive CD4 T cells to activate T helper cells, MHC class 1 molecules present antigens to naive CD8 T cells, leading to their differentiation into cytotoxic T lymphocytes. CTLs are essential for targeting and destroying infected cells, especially those infected by viruses.
Wow. Dr. Max, this is incredible. Look at this map on the screen. It's like a beautifully orchestrated symphony where every cell knows its part and plays it perfectly. The way these signals, or cytokines, coordinate everything is just mind-blowing.
I'm glad you find it amazing, Sela. This map indeed shows the complexity and precision of our immune system. It's fascinating how these tiny proteins, cytokines, can orchestrate such a robust and effective immune response. This map will be attached as a A4 printable paper for anyone to print and see at any time. We've journeyed through the intricate and fascinating world of our immune system, from recognizing pathogens to the symphony of cytokines orchestrating the defense. Understanding these processes highlights the incredible complexity and efficiency of our body's immune response.
Incredible, Dr. Max! I hope everyone found this as enlightening as I did. The way our immune system works is truly a marvel of nature.
Absolutely, Sela. And remember, staying curious and informed is key to appreciating the wonders of science.
We hope you enjoyed this deep dive into the immune system. If you found this video helpful, please give it a thumbs up.
Don't forget to hit the subscribe button and ring the notification bell so you never miss an update from us.
And we'd love to hear your thoughts or any questions you might have. Leave us a comment below.
Thanks for watching, and stay tuned for more exciting and informative videos from the Infection Tube.
Stay healthy and curious. Bye for now.